Alright, so this is a little bit more of Chapter 7, which is all about linkage, recombination, and genetic mapping. These are all really important concepts in genetics. Genetic mapping in particular is really important. You've probably heard of mapping the human genome, all that kind of stuff. And that all began with being able to map genes by doing specific types of genetic crosses, and then calculating the distance between genes on a chromosome. And so once you make a genetic map, it sort of looks something like this. You say, okay, well, here's two genes, A and B, and those genes are 20 map units apart. Then maybe there's another gene down here, C, and maybe that's 15 map units from B, or from A, and then B and C would be 35. And just so you know, these numbers don't always come out exactly so even, but that's what we mean when we mean a genetic map. We just mean putting things sort of on a line and putting relative distances between them. And that's very useful for lots of things in genetics. It's important to know where genes are if you want to study that gene, if you want to clone that gene, whatever you want to do. So genetic mapping is important. And we will apply this a little bit later to human genes and how, for example, how you can take a gene and see if it's linked to a particular disease, or to try to find out the position on a chromosome of a particular disease gene. But before we do that, we're going to work with this snail, which I won't even bother trying to say that. I don't know. So anyways, so we've got some weird symbols here. So we've got banded, which is recessive to unbanded. And there are different genes at a different locus determine the background color of the shell. So I've got yellow and brown. So we have banded yellow. So banded is big B. Well, so for sure, write it out. Banded yellow. Cross to homozygous brown unbanded. And I'm going to write that unbanded brown instead of brown unbanded, just so I have my two traits are on the same side. If you keep your banded unbanded on the left side and your color on the right side, it helps things out. So banded yellow. So banded is recessive and yellow is recessive. So if it's banded yellow, we know what the genotype is. So that means we have big B, so that's banded. We're homozygous for that. We're yellow. We're homozygous for that. We've got unbanded, which is, that's the B, O, and it's homozygous for unbanded. That's dominant, so they had to tell you it was homozygous. And then color is brown, like this. So this is one of these situations where they give you an allele designation, which is like this. This is a locus designation. So this is the C locus, and this is the BW allele at that locus. And this is the C locus, and this is the Y allele at that locus. And so they do a test cross. So that's a test cross because these guys are recessive. So banded yellow, banded is recessive, and yellow is recessive. So this is the tester. Well, it's not the tester yet, but it will be. That's what the tester would be when we did it. So if we do this, we have to end up being heterozygous for this gene and heterozygous for this gene. Then we do a test cross. So a test cross has to be banded yellow. And so these are recessive. And so we really don't care all that much about these alleles. The genotype of this is homozygous recessive, and whatever phenotype we get in the offspring is going to be due to whatever genotype we get from this individual. So what will the results of the test cross be if the loci controlling banding and color are linked with no crossing over? So that means completely linked. Alright, so now here it matters how we set this cross up. So because we started with banded yellow, we now know that banded and yellow started together, because these guys started together. And so that means we got one of these chromosomes from this parent and it looked like this. And then we got a chromosome from this parent where we had to get this and this together. And now we're saying that they're linked and there's no crossing over. And so if that's the case, all you can get is this and this. So when we do this cross, we'll end up with these kinds of gametes and these kinds of gametes. And when they get mated to this guy, these guys can only make this kind of gamete. And so we'll get, these guys are going to be, because these are recessive, because it's a test cross, the phenotype will be the same as this. So B with a big B is banded, and yellow, and BO is unbanded, and brown because of this. You'll notice up here now, banded yellow, unbanded brown. These are the parents. So these are parentals. Yeah, we'll go with green. So parentals. So we get 100% parentals. And we get 0 recombinants. So that actually, you could actually calculate that frequency as 0%. So that means these genes are super close together. So that would be our genetic distance is 0%. Alright, so I want to see if I could cut this. And then I guess that didn't work. Alright, I was trying to figure out, there's an easy way to cut that and paste it into the notepad, but I haven't figured that out yet. So our other two options we'll just have to remember for now. I'm just going to that other thing so I have some more room to write. I don't think I have any more room here. So that was if there's no crossing over, then sorting independently in 20 map units apart. I will give you a question like this on the test. This is really what it's all about. Figuring out what is going on when there's no crossing over, when the genes are sorted independently, and when they're some map units apart. If you're given these map units apart, you can always figure it out. Alright, so we're going to go with, we're going to redo this on this other thing. So banded yellow and unbanded brown. So we have banded yellow crossed to unbanded and brown. Alright, so this is banded and yellow. Banded and yellow. Cross to unbanded and brown. Unbanded and brown. So these guys are homozygous, so they have to, so we put this line here because we're just showing how the genes start out together. And so this is the parent generation. So, and then this is just the genotype of the parents. And then we can get this chromosome and this chromosome. And we get this. And we get this. And now we showed that if there's no crossing over, we just get parentals. Some kind of weird color scheme going on here, but that's all right. So what happens if we get independent assortment? Well, these things are always asking what is going on in this parent over here. So we have our double heterozygote, or a dihybrid, if you will. So we're actually worried about what's going on with this guy. This is our heterozygote. And actually, if there's independent assortment, we would draw it like that. And you might put a semicolon in there just to show that they're unlinked. And so if I got to choose from these colors, I might as well go with bright pink. And so now we can get these different types of things. We can get these together. We can get these together. Now these are our parentals. We know there are parentals because we know that that's how we started out. We know where the crosses started. So that tells us what the parents are. But we can still get recombinants. In this one. These are the recombinants. But they'll all be the same. 1, 1, 1, 1. Because this question asked about independent assortment. So P equals R, or the recombination frequency equals 50%. All right. But so now what if we've got a heterozygote and it was like this? Now they're linked, but they're 20 map units apart. What does that mean? Well, we can get our parentals. We can get the other kind of parental. We can get our recombinants. We can get our other kind of recombinants. And when I say recombinants, I'm saying that we get this one and this one, and we get this one and this one. And now they're actually due to crossing over, so that's why we draw that X in there. So we can draw it like that. These are the parentals. These are the recombinants. 20 map units apart, that means the recombination frequency equals 20%. That means each recombinant is 10%. And our parentals, that's 20%. There's 80% left over. And each parental is 40%. And so that would be the predicted offspring if the genes were linked and 20 map units apart. And then the phenotypes of these guys would be banded yellow, unbanded brown, banded brown, and then these guys would be unbranded and yellow. And these guys, banded yellow and unbanded brown, they were the original parents. So the original parents were banded yellow, unbanded brown. So these were the parents. So now banded yellow, unbanded brown, those are the parents. These are the recombinants. And that's how you do mapping. Alright, so here's another common type of problem you see with genetic mapping. These are pretty straightforward. Really get some practice with these, and you can actually, this one, if you get really good at it, you always have to be careful and not jump to too many conclusions, but you can see that you've got this kind of a pattern where you've got, without even doing anything, without even looking at the cross. I mean, especially if you kind of know this kind of thing is coming. What you can see is that you got 418 here, 426 there. They kind of go together. 19 and 16 go together. Now, if this is a test cross, and my goodness, look at that. It says it right there. It's a test cross. And these are the progeny. You know exactly what's going on. We know, because it's a test cross, we know that even before we know what anything is about which ones are supposed to be the parentals or anything like that, we know that the parentals are the 418 and the 416. And the recombinants are the 19 and the 16. So we know that. And so we know the percent recombination between these genes is 19 plus 16 over the total. And that is, so the total is 418 plus 426 plus 19 plus 16 equals 879. So that's the total. So 19 plus 16 is 35. 35 over 879 is 0.039. But we have to do that in percent, so it's 3.9%. So whatever these gene are, we got the red eyes and white banded. That's our genetic map, 3.9. And so 3.9% is also map units or centimorgans that can also be used as the units. So that's the answer to that. Now if we look at it a little more closely, we've got red eyes and banded wing are encoded by 2 mutant alleles that are recessive, so it's always important to know which ones are recessive. The 2 genes are on the same chromosome. A moth homozygous for red eyes and banded wings, so we're saying we'll just give it R and B. Red and banded is crossed with a moth homozygous for wild type, so plus and plus. So that's the cross. But then that gives us this, and then this is what we test cross. And then we take that and we test crossed it to something that's homozygous recessive. And then wild type is a parental, and red banded is a parental, and then these other guys are recombinants. So this was a 2. So 418 was wild type. It's wild type for both genes, so that's this chromosome. The other one was red and banded, so that's the other chromosome. The 19 is just red, so it's red and wild type for the other one. And this is plus and banded. So those are the recombinants. Then it says what phenotypic proportions would be expected if the genes are on different chromosomes. That means they're unlinked. what phenotypic, so if they say proportions, then you just have to worry about just sort of the fractions or the decimals. So it'll be 1 to 1 to 1 to 1. And if you wanted to come up with numbers, you could use this 879 times 0.25 or divided by 4, and you would get 219.75. So if you did a chi-square on this and you expected 1 to 1 to 1 to 1, these would be your expected numbers. But it didn't really ask you for that. Alright, so that's all there is to that. Let's do it again. So we had red banded, red and banded, crossed to wild type. And then we get an F1 that's going to be all wild type because red and banded are recessive. Do a test cross to something that's red and banded. And then we got 418, 426, 19 and 16. And these were wild type, red and banded, red and banded. Now these might be a little bit off. I don't want to go back and look precisely at what they were. I just want to take another look at this one more time, just to put in the genotypes. So red and banded, wild type. Makes this F1 heterozygous for red and banded. It makes this guy, he's got to be homozygous for both because it's a test cross. Now these are the parents. We know these are the parents for two reasons. One, because they look like the parents. And these are the parents. These not parents. Right? So we're talking about the parental generation, then the F1, then we do a test cross. So the parents are red and banded, and wild type. Wild type. So that means the recombinants are the other ones. So if you're given the parental cross, that tells you what the parents are. Sometimes you'll just say, we've got an F1 that's doubly heterozygous for something. We do a test cross, and we get these results. When you get these results, the two categories that are the most, those are the parentals. The other ones are recombinants. The frequency is 19 plus 16 over 879, which turned out to be 3.9%. All right, let's look at this. So now that we're going to cross red, cross 2 banded. So we're going to do it a little bit differently. We need a new page for that. So let's cross red, cross to banded. We're going to get all wild type in the F1. We're going to do a test cross of red and banded. What will we get? Well, we will get some wild type, some red banded, some red, and some banded. Now, which one of these are the parentals? Well, that's the parental generation. Our parental ones are the red ones and the banded ones. So parental, parental, which makes these guys recombinant. We know that the difference, or the distance between these genes is red banded is 3.9%. So that means our recombination frequency is 3.9%, which means that the recombinants are going to be half of this. So half of 3.9% is 2. What is it? Let's see, I'll have to. So 3.9, come on, calculate over here, divided by 2 is 1.95. So we'd expect 1.95%, 1.95%, and then 100% minus 3.9% is 96.1% is our parentals, but we need half of those, so it's 48.05. So that's what we expect if we do the cross this way. Because these guys, the genotype of these guys are red, but they're wild type for the other gene. These guys are banded, but they're wild type for the other gene. So these guys are heterozygous like this, and these are in repulsion. The previous example, they were coupled. So now the red and the banded are our parentals, because that's how they started out. So think of it as red plus and plus banded, and then these guys are plus plus, and these guys are RB. So these guys are like red plus. These guys are plus B. So you can do it in different ways. All right, let's do another one. In cucumbers, we got some heart-shaped leaves. So we'll just, again, you can make whatever you want. I'm not going to go with HL every time, so we'll go with little h and big H. And we've got numerous spines and few spines. So little n is numerous, big N is this. So we got little h is heart-shaped, big H is normal, and little n is fruit, is numerous, and big N is few. Alright, so whatever all that means. But if we use little h and little n, then that tells us which one's dominant, which one's recessive. The genes for leaf shape and number of spines are located on the same chromosome, so that means they're linked. Mapping experiments indicate they're 32.6 map units apart, so that's important. So now we're going to take heart-shaped leaves. Heart's recessive, so if they have heart-shaped leaves, they're little h, little h. Heart-shaped leaves and numerous spines. And numerous is recessive, so numerous and numerous is crossed with a plant that's homozygous for normal leaves and few spines. Alright. The F1 are crossed with plants that are heart-shaped and numerous. Alright, so what are we going to get out of this? I'm digging this color. I don't know, I tend to like blue for some reason. Well, let's use green because these are plants. Alright, so we can get this chromosome. And we can get this chromosome. Doesn't matter when you do it like this which ones are on top and which ones are on bottom. Then we do a test cross. Now we know these are the parents, so these are the parent generation. We know how we started with the parent generation. This is the F1, and then this is the tester. So again, these are not the parents. These are the parents. So we're going to get, we know what our parentals are. Little h, little n, big H, big N, those are the parents. And then from up, then the recombinants are going to have to be, if we go from so where do the recombinants come from? The recombinants come from if we get crossing over between these two. And so now H is going with N, little h is going with big N, big H is going with little n, little h, big N, big H, little n. These are recombinants. These are parentals. Map distance is 32.6. 32.6 divided by 2 is 16.3. So this is percent recombination. So 16.3, you get 16.3 of these, 16.3 of these. 100 minus 32.6 is 67.4. So 100 minus 32.6 equals 67.4. Half of that is 33.7. So that's what you expect. So you can do these problems in two ways. You can either be given the numbers, be given numbers like this, and then use that to calculate your recombination frequency, or you're given the recombination frequency and asked to come up with the predicted results of a cross. Alright, so here's a similar thing, and we're just kind of looking at it a different way. We're doing a test cross, and we're getting, then they're giving us these results. We got two different sets of results. So they're essentially asking, so what we have is these two genes, and so we're doing a test cross for that's going to look like this. So if you read this whole thing, farmer has two tall, smooth tomato plants, and they both are heterozygous, but he doesn't know how they're heterozygous. And there's two different ways it can be heterozygous. It can be like this, or it can be like this. Where this is a little p. This is coupled, and this is in repulsion. Because here the big D and the big P are on opposite sides. Here the big D and the big P are on the same side. So which kind of plant, so how do I know all that? Up here we have a farmer has two tall is dominant, and smooth is dominant. And then he does a test cross, crosses plants A and B with the same dwarf pubescent, so dwarf and pubescent are both recessive, so that's the test cross, so that's these guys. And he gets something that looks like this. And because you get that, that fits the pattern of a test cross between two linked genes, where you get a lot of two things and then fewer of the other two things. And then here we just have different. We have high numbers and low numbers, but the things that are high and the things that are low are a little bit different. So if we do this first one, if we look at these, the first one, because this is a test cross, we're asking what came from this heterozygous parent. And we can get big D and big P, we can get big D and little p, we can get little d and big P, we can get little d and little p. But from this tester, we can only get little d and little p every time. And so this little column here and this column here with the little d and the little p, those all came from the tester. The other d's and p's came from the heterozygote. So plant A, big D and big P are starting out together. So plant A. Because you get more of big D's and big P's, and then little d and little p from that parent. So that means those are the parentals. And if those are the parentals, that means it started out like this. And then we get recombinants like this. The other one our parentals are these guys, parentals. These parentals have a big D and a little p, and a little d and a big P. So big D, little p, little d, big P. That's how we start out. So in plant B, we start out that way. So our parentals are, make this a little p, got a big P. So these are our parentals. And then we get a crossover. We get big D, big P, and little d, little p. These are recombinants. Alright, so are these genes linked, and what's the distance between them? Yes, they're linked. And we know that because if they weren't linked, we'd get a 1-to-1-to-1-to-1 ratio. And also, if they weren't linked, it wouldn't matter how there's independent assortment, so there's no repulsion and coupling. You just get a 1-to-1-to-1-to-1 ratio. Alright, so let's calculate this out. In the first case, we've got 2 plus 4 over the total. And so in plant A, we've got 122 plus 124 plus 6 plus 4 equals 256. So this comes out to 6 divided by 256, and that's 2.3%. I'll leave you guys to do those calculations. I suppose I should have used a different color there. But we'll switch up our colors just to make it look different. These guys, we've got, oops, no, this was 6 plus 4. So this guy was 6 plus 4. So 6 plus 4, and then we add all those up. So it's not going to be that much different, but we'll see what it is. 122 plus 124 plus 6 plus 4. So that's still 256. But now we've got 10 divided by 256. That's 3.9%. Oh yeah, that's kind of a lot. So that was 3.9%. Kind of coincidental that other cross also had 3.9%. And then this one is 6 total over, so these 6, 2 plus 4, over, I can do this in my head, 164 plus 6 is 170. So 6 divided by 170 is 3.5%. So that seems pretty close. Oops, not sure what happened there. Randomly closed on me. Glad all my writing's still there. So 3.5%. 3.9%. So these are easy. Just practice those. I could give you 50 of them on the next exam. You should still have time to do them. Probably won't give you 50. Alright, here's another one. A and B are at a locus. So this is just kind of getting abstract. Not talking about specific traits or anything like that. And what kind of conclusions can we make? So it's saying, so here we're saying we have this kind of a heterozygote. But that doesn't give you enough information. All that does is tell you that it's heterozygous. It could be like this. It could be like this. This is coupled. This is in repulsion. This guy will give you these kind of parents and these kinds of recombinants. these guys will give you these kinds of parents and these kinds of recombinants. So which one is it? Well, these are the parents because there's more of those. These are the recombinants because there's less of those. How do we interpret these things? Well, the little a and the little b came from the tester. The big A and the big B came from the heterozygote. So the cross is this. So a little a and a little b came from this one. So we can essentially cross out that B and that A. And then the things that came from here are big A, big B. So the thing here that came from the heterozygote are big A, little b. We got more of those. So which one is it? It's this one, because this is the parent. And we got 45 of these, 45 of these, 5 of these, 5 of these. $10 divided by 100 equals 10%. So then we would have a genetic map. We would just say these are 10 map units apart. And it doesn't matter here when you're making your map. It doesn't matter if you use the big A, the big A here, or the little a, or the big B here, or the little b here. We're saying that the genetic locus is 10 map units apart. And the genetic locus is just called the A locus. This one's a little bit different. And it's starting to look at real data. Whenever you start looking at real data, it gets a little bit, it just makes it a little bit less obvious, I guess. And the numbers are a little wacky. But now this is how we would do, and you can tell, they show you the Y chromosome there, so you can tell it's X-linked. And so the X-linked mapping is a little bit different than the mapping of the autosomal genes. So just looking at this, you can tell that it's X-linked because they're showing the Y chromosome there in the male. But they're not showing the X chromosome because, they're not showing it in this kind of fashion. Just because it's hard to interpret what's going on with that X chromosome. So we just sort of ignore that. Alright, so Daniel McDonald and Nancy Peer examined the eye spot in flower beetles caused by an X-linked gene that's recessive to the allele for the absence of eye spots. And then they did a series of crosses to determine the distance between the gene for eye spot and a dominant X-linked gene for striped. So this one's a little weird because now we got striped is the mutant allele and it's dominant. So in this case, the wild type allele for striped is going to be recessive. And it acts as a recessive lethal. Oh, so this one's hard. Alright, so this has got a lot going on. So let's think about that one for a minute. So we got our big S T gives you striped. But big ST over big ST is lethal. ST plus is fine, and it's, I guess, not striped. And that's wild type. I spot is just normal recessiveness. So here, this is a test cross because, well, it's kind of like a test cross. This guy is heterozygous for both genes. So that's good. We want to be able to we want to show when recombination has occurred. So we really only care about what's happening in this individual. So we're only interested in the gametes produced by that individual. Then we're going to cross that to this male. But the problem is we can't make the male recessive for the striped phenotype, or can we? Well, yes, we can. So it's because the striped is dominant, so it's not as hard as I thought. So striped is dominant, and this ST plus is so the ST plus is recessive. So wild type is recessive. The reason I'm hesitating is because you can tell there's a big S there, and that makes you think that it is dominant. But the way they name these mutant alleles is because the initial mutant is dominant, has a big S. The mutant allele is called big S, and this in that big ST over big ST is lethal, but big ST plus, that's the recessive. So these guys are recessive. So ES is recessive and ST plus is recessive. So we've got ES plus ST over ES ST plus. Alright. So the types of gametes we can get parentals. So this is going to be a parental because it's like that. This is going to be a parental. So it's like that. We can get this. These are the types of gametes we can get. So that's a crossover here with ES and ST. And this guy is ES plus and big ST plus. So that's what's happening in our females. And then the types of gametes that we can get from the other parent are, so that's from this parent, are ES and big ST plus, or the Y. So we can get all these things. But ST over ST is lethal, and if it's a recessive lethal, that means also that ST over Y is lethal. Because if it's recessive, then if it's over Y, you still get the same phenotype. So big ST, whenever you get that, which is here and here, over Y, we don't get those. So we won't see any big STs over Y over here. So we don't see those. So we'll keep that in mind. Alright. And so now we know that the way they've written this out, that from this parent over here, we can get ES and ST plus. So all these guys down here are from the tester. And then these are from the tester. So the way they wrote it out, the alleles on the bottom of the line, below the line, are from the tester. And then what's on top of the line are what we get, are the gametes that we get from this parent over here, the one that's circled in blue. Over here we know this is a parent, this is a parent, this is recombinant, this is recombinant. We know that because of the way they set up the cross. They actually gave us how they set up the cross. So we know that this is a parent configuration and this is a parent configuration. So anything with an ES plus ST on top is a parent. So that's a parent, ES plus ST, and that's the only parent of that kind that we've got, because there's no other ES plus ST. ES ST plus, this one's a parent. This one's a parent. Okay. ES-ST, that's down here, that's recombinant. ES plus ST plus, that's recombinant. ES plus ST plus, that's recombinant. So what are we missing? We're missing these guys and these guys. So that's ES plus ST over Y and ES ST over Y. So we're missing a parent and we're missing a recombinant. And by the way, these are females. These are females. These are males. And so we're missing two classes of males. So this ES plus ST is the same as this ES plus ST over ES ST plus. So these males that get this ES plus ST should be the same frequency as the ones that are ES plus ST over ES ST plus. In other words, these females here, we should get this chromosome equally, this chromosome here, we should get equally frequently over the Y chromosome, but they die. So if we're going to calculate all of our parentals and our recombinants, we could do a couple of different things. We could just assume that we got the same number of parentals in the males, and we could just add another group of parentals that's similar number to that. And similarly, we can get these recombinants, ES-ST, those are like these guys. We could add those in as well. We could add in the male versions of those and add them all up. But what we can also do is just say, because we're missing half of the males, and we really just want a percentage, we can really just think about what's going on in this half of this Punnett square over here. Or in other words, we can just ignore the males and take a look at the females. So if we just look at females, we get one of those situations where we got the parentals are more than the recombinants. Did I mention this was a hard one? And so we just add them all up, just these guys. So we would add 1630, 1665, 935, 1005. That's the total. The recombinants are 935, 1005. And so we just add all that stuff up. So 935 plus 1005 is 1940. And then 1940, because I don't have to add those up again, plus 1630 plus 1665 is 5235. 1940 divided by 5235 is 37%. So that's the distance between those. So that's pretty goofy. But if we added in another set of numbers, so we could add in another 1630 that would look like that, but would be males, and we could add another 935 that would look like that and would be males, and we'd get the same number, because we'd be adding to the top and the bottom. So anyways, that was a crazy one. You certainly won't get anything that hard on the exam, but it's a good example of where you can look at data and you can figure out what's going on, even if it is complicated. And the key is, what we want to do is we really just want to look at what's going on in this heterozygote. We just want that heterozygous female, and we want to know which ones are the parents, which are the recombinants, and count them up and calculate a percent and go from there. Alright, so that's a tough one. A tough one. I mean, that's why we do them for practice. Well, if we go for a tough one, we might as well do an easy one. So this one's pretty easy. So these are recombination distances. So these are just saying that they did test crosses between these different genes in corn, and they got these recombination frequencies. So another way to write that would be that between these two genes, there's 17 map units. Between these two genes, R and L2, there's 35 map units. And between W2 and L2, there's 18 map units. So we need to put this together into a map. And so we know that R and W2 are 17 map units apart. Then R and L2 are 35 map units apart. So I'm going to move this one over so we can have some room. So R W2 is 17. And then we'll write another possibility over here. R W2 is 17. And then we say R and L2 is 35. So L2 could be over here. So these are two different maps, they're not related. Let's put this other option down here so I have more room. So over here we could have L2 could be over here. Because we know that R and L2 are 35 map units apart. If R and L2 are 35 map units apart, then this would be 18, which is W2, L2. And that's consistent with this. So this map is consistent with those numbers. But another possibility before we actually do this is that RW2 is 17, RL2 is 35, but then L2 and W2 would be 35, 45, 52. So they'd be really far apart. So then we would expect this number to be like 50%. And so our better map is RW2, L2 with distances like this. So that would be our most consistent map. Easy enough. Those ones aren't too bad. Alright, so here's another one. This one, you are given the map units ahead of time. So that means our number of recombinants is going to be 7%. Or we'll have 3.5% of one type of recombinant and 3.5% of the other type of recombinant. So all we have to do is figure out the types of recombinants in each case. Alright, so this one, so these are all, oh boy, these are hard. So these two are easy. Top two are easy. So these are test crosses. And so in this test cross, we got D and OP are the parents, and little d and little op are the other parents, because that's how they start out together. And then big D, little op is a recombinant, little d, big op is another recombinant. So parent, parent, recombinant, recombinant. 3.5%, 3.5%. 100 minus 7% is 93%, so that's 46.5%. Nope, 93, so 93 divided by 2 is 40, Is it 46.5? No, it can't be. Alright, hold on, I gotta do the math. So 100 minus 7, that part I knew. 93% divided by 2. Yeah, 46.5. Just didn't seem right for some reason. So 46.5 adds up to 100%. This guy is, now we've got big D, little op, little d, big op. Those are our parentals because of the way they start out together. Parentals. Recombinants are going to be, if we look at this guy over here, big D to big op, little d to little op. Big D, big op, little d, little op. These are recombinants, so there are different things that are recombinants here. So these are big and small, both of them. And so this would be 3.5, 3.5, 46.5, 46.5. Or if we wanted to put it up here just to compare it directly, we would switch these. So we'd have 3.5, 3.5, 46.5, 46.5. That's what this cross would give you. And that's what this cross would give you. Now these guys are not test crosses because we've got dominant alleles coming in from the other side. So these are in fact both dihybrid crosses. Well, we don't have anything better to do. Let's take a look at what's going on there. So we need a new page. And we've got D. They did it in two ways. We'll just do it in one way. So what happens if we do a dihybrid cross? So this is, I always say this kind of a problem is good to demonstrate why we don't do dihybrid crosses with this. So we know what happens with the gametes from this. We know that these are the parents. I wish they give us better letters that we could see big and little. And then little d, big OP, and big D, little OP. So just like before, if these are the parents, this is 46.5. This is 46.5. This is 3.5. This is 3.5. The problem is, now we have to kind of do a Punnett square is one way to do it, at least one way to demonstrate it. Big D, OP, little d, little OP, big D, little OP, and little d, big OP. But we also get, and now in this we get the same thing. We get 46.5, 46.5, 3.5, and 3.5. So this is for the advanced people that like to think about things a little deeper. It's not that hard, really. But now it's just saying what is the probability of getting this genotype. And if we get these guys at this frequency and these guys at this frequency, these guys here are going to, take, when you multiply percents, you take 0.465 times 0.465, which is 0.216. So you get 0.216 of these. And then you have to calculate exactly the probabilities of all these other things. And so now we have, let's say for example, big D OP and little d OP. That would be 0.35 times 0.35 0.1225. So that would be 0.1225 would be the probability of getting that genotype. Then you'd have to add up all the ones that give you the dominant phenotype and all the ones that give you the recessive phenotype. And let's see, so the recessive phenotype in this case, well, they're more common. So I'm not going to do all that math. It's kind of ridiculous. But you can, what you're doing is calculating the frequency of this, calculating the frequency of this, and then multiplying each frequency by the other to calculate the frequency of getting them both together. It's the same thing. If these things were unlinked, then we would get 1 to 1 to 1 to 1 to 1 on both sides because of independent assortment. And when we filled in all those boxes, we'd get 9 to 3 to 3 to 1. But if they're linked, we don't get that. And that's why if you do a virtual fly lab cross and you try to do a dihybrid cross with linked genes, everything is all messed up because you have to, depending on how far apart they are and exactly how the cross is done, it gets pretty goofy. So now I will put tricky stuff on the test, but probably not that tricky. We need this to go away. Go away. There we go. Alright, so those are tricky. I won't bother with the other one. This one, German cockroaches with bulging eyes and curved wings, recessive to straight wings. Both traits are recorded by autosomal genes that are linked. Oh, this is an easy one. Multiple choice. Man, I guess that last one was so hard they wanted to give you an easy one. So they're just asking you if you know what this term repulsion means. So we have something that's heterozygous like this. Genes are in repulsion. So we're going to have BU plus CV. And over here we'll have BU CV plus. So that just means that the same kinds of alleles are on the opposite side of the line and they're going in different directions. And same thing here, these guys are going in, so these two are going in different directions. So we're going to get, if they're in repulsion, we're going to get these two things more frequently. So BU plus CV is one of them. That's not an option. BU CV plus, that is an option. So of all these other guys, this one will be most frequent. You'll never get these, you'll never get these. BU CV is a crossover, and BU plus CV plus is a crossover. So that would be recombinant, that would be recombinant, that would be a parental. There's no way you can get BU, BU. Well, in order to get BU, BU, you'd have to get some kind of a mutant and lose that, or there's some other things that can happen. And then it didn't even show you the CV. So those other two are not really options. Alright, so those are all the things with 2 genes. Then we're going to start doing things with 3 genes. So we'll leave this here, and then we'll start looking at 3 genes. Now in this case, these 3 genes aren't linked because they're showing you here that that line's not connected. So we're talking about 2 linked genes and 1 unlinked gene. I think we can do this one. So we'll do this one and then we'll leave it at that. So I'm going to get a slightly smaller pen here so I have more room to can squeeze more stuff in there. Alright, so we can do this. This is easy enough. And so, we've got these 2 genes. What do we got here? Ebony and Rough Eyes are on chromosome 3, separated by 20 map units from forked bristles. Well, they're separated by 20 map units from each other. And then forked bristles is on the X. And it sorts independently of E and RO. Of course it does, because E and RO are on the 3rd chromosome. And forked is X-linked. So that means they're on different chromosomes. If they're on different chromosomes, they sort independently. Give the phenotypes of progeny and their expected proportions when a female of each of the following genotypes is test crossed with a male. And I just gave you one So it said each of the following genotypes, but I just thought I'd do one example. Alright, easy enough. So what do? We calculate each thing independently. So we have to do a test cross, so it's got to be ERO over ERO, and then it's going to be a male, so it's going to be forked over Y. And so it's going to be the kind of thing where we just care about what's happening in this female and what kinds of gametes are getting produced. So let's think of these kinds of gametes by themselves. These are the parentals. These things are 20 map units apart. And then we could also get this. These are our recombinants. So recombinants, 20 map units apart, means each one of these is 0.1. 0.1 and 0.1 is 0.2, so each of these is 0.4. Or 40%, 40%, 10%, 10%. We know that because the number of recombinants equals 20 map units, which is 20%. Then each recombinant is 10%. So that's how I got this. These guys are going to be 1/2 forked plus and 1/2 forked. And if you get forked plus, you can either get an F or a Y, and you're just going to be wild type for forked. If you get a little f, you can get an F or a Y, you're going to be mutant for forked. So the males and females are going to give you the same answer here, so we can ignore that. So our options now, we're going to have 8 options because we have these 4 options, and then we can combine these with these things. So we can have E plus, RO plus, ERO, E plus, RO, ERO plus, and these are 0.4, 0.4, 0.1, 0.1, and then of these, 0.5 of these are going to be forked plus. So how many are going to be how many of these are going to be all 3 of these things? Well, 0.4 times 0.5 is that 0.02 or 0.2? 0.2. So 0.2 will be E plus R, we'll just go R plus F plus. 0.2 will be E R F plus. 0.1 times 0.5 is 0.05. E plus R F plus. 0.05 E R plus F plus. And then you'd have the same numbers for the other things. So the other half would be E plus R plus E R, E plus R, E R plus. And then all these guys were also F plus. All these guys are going to be F. But we'll get the same frequencies. 0.2, 0.2, 0.05, 0.05. If we add all those up, 0.2, 0.2, 0.2, and 0.2 is 0.8. And 0.05 and 0.05 and 0.05 and 0.05 is 0.2. So it all adds up to 1, which is 100%. So I went through that kind of quickly. But it's the same principle as before. If you know what's going on just for a couple genes, for one cross, calculate what's going on for that gene, calculate what's going on for the other gene, and now we've got independent assortment because what's going on with these guys is independent of what's going on with forked. So if there's independent assortment, then we can multiply those probabilities together. Okay, one more. Because I want to stop when we get to this kind of cross. I want to stop when we get to the 3-point mapping. This is making for kind of a long... Alright, we'll stop here, because this one's too long already. So we'll stop here, and